So, have you ever stopped to think about how a single, perfectly normal cell in our body can just go rogue? How does it decide to rebel against all the rules and become cancerous? Well, that's exactly what we're going to do. We're going to take a visual journey, step by step, and map out the entire process from that very first mistake all the way to a full-blown invasion.
Just take a look at this. On the left, you've got this beautiful, orderly community of cells, all working together. And on the right, it's just chaos. So how does this transformation happen? Understanding this journey from cooperation to complete anarchy is really the key to understanding cancer itself.
All right, so here's our game plan. We're going to break this story down into 6 key parts. We'll start with that initial spark of rebellion, and we'll follow it right up to how doctors actually see and describe it in a clinical setting. Let's dive right into part 1.
Every rebellion has to start somewhere, right? For cancer, that spark happens deep inside a cell, in its most fundamental rulebook, the DNA. It all kicks off with a series of genetic mistakes. You can think of a cell's life cycle kind of like a car. You've got an accelerator. These are called proto-oncogenes that tells the cell, okay, it's time to grow and divide. And you've also got brakes. We call those tumor suppressor genes that say, whoa, slow down, stop, or let's fix this damage. Cancer begins when something breaks this incredible fine-tuned system.
Let's zoom in on one of those brake pedals, a gene called P53. This one is so ridiculously important for preventing genetic screw-ups that scientists have nicknamed it the guardian of the genome. And trust me, it's one of the most powerful defenses our body has against cancer. So here's the play-by-play for how P53 works. Let's say a cell's DNA gets zapped by some radiation. P53 senses that trouble immediately and just slams on the brakes, stopping the cell from dividing. This gives the cell a chance to make repairs. But, and this is crucial, if the damage is just too severe, P53 makes the tough call. It triggers something called apoptosis, which is basically programmed cell death. It tells the cell to sacrifice itself for the greater good of the body.
Okay, so that's what's supposed to happen. But what if the guardian itself is mutated and broken? If that brake pedal is busted, then damaged cells don't get stopped. Their DNA doesn't get repaired, and instead they just keep dividing, passing on all those dangerous errors. And that's exactly how a tumor can get its start.
But it's not just about broken brakes. Sometimes the accelerator gets jammed to the floor. This is a process called gene amplification. So a normal cell, like the one on the left, has just 2 copies of a growth gene. But look at the cancer cell on the right. It's made hundreds of copies of that same gene. I mean, can you imagine 100 accelerators all floored at once? That is the kind of relentless, out-of-control growth we're talking about here.
So let's move on to our next section and see where this is all going. When these mutated cells start dividing like crazy, they form a mass, or a tumor. But here's the thing. Not all tumors are created equal. And this is a really critical distinction we need to make.
This chart really breaks it down for us. A benign tumor is kind of like a well-behaved outlaw colony. It's usually contained in a capsule, it grows pretty slowly, and it stays put. But a malignant tumor, now that's cancer. It's not contained, it grows fast, and, most importantly, it invades the tissue around it and can metastasize, which means it can spread.
And here is a perfect picture of what I mean. This is a benign tumor, a leiomyoma. Just notice how the cells still look a lot like normal smooth muscle cells. We call this well-differentiated, which is just a fancy way of saying they still remember what their job is. They look pretty organized, almost civilized.
Malignant cells, on the other hand, undergo something called anaplasia, and this is a super important concept. It means they completely lose their differentiation. They totally forget who they are and what they were supposed to do. They become pleomorphic, meaning they're all different shapes and sizes. These are the true rebels, and they look nothing like their law-abiding ancestors.
So as these malignant cells evolve, they don't just grow. They start picking up a whole set of devious new abilities, what scientists call the hallmarks of cancer. You can really think of them as superpowers that help the tumor to not just survive, but to thrive.
So here are some of those core superpowers. They learn how to flat out ignore signals that tell them it's time to die. They figure out ways to become invisible to the immune system, and maybe the most incredible, unbelievable superpower of them all, they find a way to become immortal.
So how on earth do they do that? Well, our normal cells have a built-in aging clock. Every time they divide, these little protective caps on our chromosomes, called telomeres, get a tiny bit shorter. Eventually, they get too short, and the cell just stops dividing. But cancer cells, they find a way to turn back time.
They reactivate an enzyme called telomerase, which rebuilds those telomeres, basically giving them the ability to divide forever. They literally become immortal.
Okay, so you've got this immortal, rapidly growing tumor. It has one big problem. It's starving. To get any bigger than a tiny speck, it needs oxygen and nutrients. So its next move is to actively hijack the body's resources and recruit its own supply lines.
What you're looking at here is an absolutely pivotal moment in the cancer's life story. On the left, you've got abnormal cells, but they're still contained. We call this carcinoma in situ. They haven't crossed this critical border called the basement membrane, but then they break through. That moment, that crossing of the border, that is invasion, and that's what makes a tumor truly malignant.
Now, once they invade, these tumors need to build infrastructure. They do this through a really sneaky process called angiogenesis, which just means creating new blood vessels. The tumor sends out chemical signals that are basically screaming at the body, hey, we need new blood vessels, right here, right now. And these new vessels don't just feed the tumor, they also create the perfect escape route.
And that escape route, well, that brings us to the most lethal characteristic of cancer, its ability to spread, or metastasize. This is the process that turns a local problem into a systemic, body-wide disease.
This journey is a treacherous one for a cancer cell, and we can break it down into 5 key stages. First, it has to invade locally. Next, it has to do what's called intravasation, squeezing into one of those new blood or lymph vessels. Then it has to survive floating around in circulation, hiding from immune cells. Then it performs extravasation, getting out of the vessel. And finally, it has to colonize a new organ, setting up a brand new outpost.
And let me be crystal clear about why this is so important. It is not usually the first original tumor that's fatal. The overwhelming reason that people die from cancer is because of metastasis. This whole cascade is what makes the disease so incredibly dangerous.
All right, for our last step, let's connect all this pathophysiology back to the real world, back to medicine. How do doctors actually describe and categorize this whole journey we've just visualized? Well, they use a system called staging. One of the most common systems is called TNM. The T tells you about the primary tumor's size and how far it has invaded locally. The N tells you if it's spread to nearby lymph nodes. And the M, which is the most critical factor for prognosis, tells us if the cancer has metastasized to distant sites. It's really just a clinical shorthand for the entire biological story we've just walked through. So, we followed this renegade cell all the way from its very first genetic mistake to setting up a new colony in a distant organ. Visualizing this step-by-step progression gives us such a powerful framework. And it leaves us with the most important question of all, right? Now that we can actually see the path it takes, where are the best places to build a roadblock?